Hello everyone, welcome to today's class. In this session, we will begin our discussion on inheritance biology. It comes under Unit 8 of the CSIR NET Lifescience syllabus. So, inheritance biology can also be referred to as genetics. This unit is divided into 6 parts. First is the chromosomal and extra chromosomal inheritance. Next part is genes and mutation. Then we'll learn about genetic analysis. Then comes human genetics. Then is quantitative genetics. And the last part is structural and numerical alteration of chromosomes. These are the major 6 parts that we will be covering in this unit. In this lecture, we will be discussing about the first part, that is chromosomal and extra chromosomal inheritance. Alright, as we all know, the human body consists of DNA, that is deoxyribonucleic acid. Like it carries all the genetic instructions required for like development, functioning, growth, reproduction of living organisms. Now, if we look at the structure of DNA, it's something like this. Like this. Like it has a double helix structure which resembles like twisted ladder, and it's formed by two antiparallel strands of nucleotides. Okay, so like if we take suppose this part, something like this. Okay, like if we analyze this part, so these are like strands. Okay, and suppose A is bound to T. These are like these are hydrogen bonds. Okay, and C is come to D, and this thing continues. Like again there may be another G, C, or a T, A, and this thing continues like entirely. So now let's discuss what is a gene. Okay, suppose if we consider the DNA, okay, like if we consider a DNA strand like this, and if we take a small part, okay, like this part, a segment of the gene, a DNA, this segment is called gene. So from a entire DNA, if we take a segment, it's called gene. So as I discussed here, the monomeric unit, so this is the DNA, right? So its monomeric unit is a nucleotide. So if we consider a gene, its monomeric unit will also be a nucleotide, right? Okay. Now let's consider a gene. Gene. So this is a DNA, and from which we take apart the gene. Okay. So we all know that DNA undergoes a process to copy itself called replication. This is not required. Okay. And this gene undergoes a process called transcription to form RNA, rather mRNA. And this mRNA undergoes another process called translation to form protein. Okay. These two processes will be discussed in mol bio. And the expression of a gene, like the gene till the protein, like if the protein formation has taken place, we call that gene expression. That is expression of a like segment of DNA to its protein, it's called gene expression. One thing you must keep in mind is that replication takes place on the entire DNA, while gene expression takes place on a segment of DNA, that is gene. Okay. So this primary flow of genetic information within a biological system, that is from DNA to RNA to protein, it's called central dogma, and it was proposed by Francis Crick. So to summarize what is gene expression, we can say that gene expression is the process by which the information in a gene is used to create a functional gene product like we can write in bracket like typically a protein or non-coding RNA molecule. Now let's learn what are alleles of a gene. So if we go by the definition, an allele is a variant form of a gene that are found in the same place, that is locus on a chromosome, and code for the same trait but with different variation. Okay, so suppose if we take a gene and consider gene A. Okay, in gene A, suppose we have a segment A, T, G, C, C, T, G, A, and this complementary segment T, A, C, G, G, A, C, T, T, and we take another segment C, T, A, G, C, C, T, T, A, G, and is complementary G, A, T, C, G, A, T, C. So, and we name this gene B. So since gene A, rather the segment A and segment B are completely different to each other, so they are two completely different genes. Okay, so A and B are two completely different genes, but if we see a slight change in the nucleotide sequence, that is suppose A, T, and there's A, C, C, T, G, A, and in this sequence it's T, A, T, G, G, A, C, T, T. We can see that in this, between these two genes, there is only change in this nucleotide, these base pairs. Okay, like there is only change in one base pair. I'm not mentioning it's the only because change in one base pair or two base pair, but like very slight change, but not like gene A and gene B where there's a complete different sequence. But if there's a very slight change between two different sequences, then we can call them alleles of a gene. We can call them alleles of a gene. Okay, and to name them, we'll name it since this is capital A, we'll call it as gene A with small a. Okay, similarly, if there's a suppose, so change like T T A G C T T A G, and here it's like A T C G A T C, we can observe that there is a change only in this base pair. So this will be gene B, small b, and we can say from here that capital A and small a are alleles. As well as we can say here capital B and small b are alleles of a gene. Fine. Now, if we look at the definition, it says allele is a variant form of a gene. This thing I explained just now. Okay, this is the explanation for the part. Variant form of a gene that are found in the same place, that is locus on a chromosome. So now let us come to this part. So what happens is that the DNA, okay, suppose this DNA, these DNA folds itself, folds with the help of histone proteins to form chromosomes. Okay, so this chromosome, okay, inside this chromosome, the gene A, this is a segment of the DNA, will occupy a position. Okay, like gene A, this is a chromosome, and gene A will occupy this position. Now we know that human beings are diploid creatures. Okay, human beings are, not only human beings, many organisms are diploid creatures. So what does diploid mean? In diploid, di means two and ploid means set. That means the diploid creatures have a set of two chromosomes. Okay, set of two chromosomes. So if we come to a definition, you can see what is said, they are found in the same place, that is locus on a chromosome. So since in diploid organism they have two sets of chromosome, okay, so suppose this is one chromosome, sorry, this is one chromosome and this is one chromosome. These two are identical, that is homologous chromosomes, okay, or homologous pair. So if gene A occupy this position, okay, then its allele A dash will also occupy this position. This not necessarily be an allele, it can also be A as well. So, but according to definition, you can see he are found in the same place, that is locus on a chromosome. See, they are occupying the same place, there is the same locus on the chromosome. For example, if we consider like DNA B and it has occupied, say, suppose there is another, this is a one chromosome, chromosome 1, okay, let's take another chromosome like a different one. So these are homologous pairs, okay, and these are homologous, and suppose B has occupied this position, so this B, small b, will occupy this position. So these are a homologous pair, and these two are a homologous pair, and if we take these two, this one and this one, they are non-homologous chromosomes. In one chromosome, more than one genes can be present, okay, let me draw chromosomes. So these are a homologous pair of chromosomes. So suppose a gene A, it may present here, okay. Also, it's not necessary that an alternate form of a gene will be present in the two different chromosomes, okay. Suppose B is taken this position, it is not necessary small b will be present here, capital B can also be, the same gene can also be present here. Another gene, suppose say gene D, that can be present here. So like different genes can be present in the different positions on a chromosome, and this the location or position is called the loci or locus or position of a gene in a chromosome. This position is universal for everyone. Like suppose for some disease, it's present in like chromosome, say chromosome 2, and it's present in this position. So for every human being, that gene for the same disease will be present in that position only, okay. So based on the placement of chromosomes in a zygote can be in two different conditions, that is like based on the presence of gene, zygote can be heterozygous or it can be homozygous, okay. Hetero means different, homo means same. So if in a zygote, like on this chromosome, if capital A and small a are present, it's called heterozygous condition. Similarly, for capital D and small d, it is a heterozygous condition. But if capital B and capital B are in the zygote of the chromosomes, it is called homozygous condition. Therefore, what does homozygous means? If we at the definition, it's homozygous. If we make a table like homozygous and heterozygous. So homozygous is the presence of two copies of the same allele which codes for a particular trait, while heterozygous is the presence of two different copies of alleles of a particular gene. That is, here the example may be like A or T, sorry, AA or maybe AA, or maybe capital T or small t, but here it will be capital A, small a, capital T, small t, like this. Okay, and we must keep in mind that when producing gametes, these produce only one type of gamete, while heterozygous chromosomes, in heterozygous conditions, they produce two types of gametes. Okay, now let's discuss about the works of Mendel. Okay, Gregor Johann Mendel is referred to as the father of genetics. Okay, what he was working for a long time, but his works were only recognized in the early 1900s when people realized. Now what he did was that he worked with pea plants. He used pea plants for experiment. Okay, pea plants. What he used to do is that he used to cross between two parent pea plants having contrasting characters, and then he used to study their progeny, and he used to again cross their progeny, and he used to study like that. So before I come to that, let me discuss what are characters. Okay, so characters are basically, sorry, one sec. So character, it is a general heritable feature of an organism. Organism. While trait is specific or specific, not or specific detectable variant or expression of that character. Okay, so what Mendel did is that he chose pea plants with 7 different characters that had like contrasting traits. Okay, contrasting, I guess you know the meaning of the word contrasting, that is which are very easily detectable and different. Okay, so he chose from his pea plant because 7 characters that had contrasting traits. Let me give you an example. Suppose one character is plant height. So this is the character, and what are the traits for that? Tall or dwarf. So the character is plant height, and it has 2 traits. Like this observable traits, that is tall or dwarf. See, if you see the example, general heritable feature of an organism. So the general heritable feature is a plant height, and it is for its trait specific detectable variant, specific detectable variant or expression. So detectable variant or expression are the tall or dwarf. Another example may be flower color. So this is the character, and what are its traits? The flower color may be purple or white. Okay, so what Mendel did is that he chose pea plants because he could easily choose 7 contrasting characters. Okay, that is easily observable, 7 easily observable characters which had contrasting traits. Okay, so the 7 characters what he chose were: number 1, plant height; number 2, flower color; number 3, flower position; Number 4 was seed shape. Number 5, seed color. Number 6, pod shape. And number 7, pod color. Okay, so these are the 7 characters Mendel chose to carry out his experiments. Okay, one thing you must keep in mind that Mendel did not name this as genes. Mendel used the term called, he used to refer these genes as factors. Okay, he used to refer to them as factors, and he proposed that these factors are the discrete units of heredity that are passed from parents to offspring, and these factors only determine the specific traits, and they exist in pairs within an individual. Okay, so he said that these factors are passed on from parents to offsprings, and these carry those specific traits which an individual expresses. So suppose we take a specific character. Okay, suppose we take plant height. So plant height has two traits, tallness and dwarfness. Okay, so you may know the term genotype and phenotype, but let me explain it again. So these which trait that will be expressed is coded in the gene. Okay, the trait, specific trait for the character that will be expressed is gene coded. Okay, which is not observable. This gene is not observable to us. It's a genetic code. It is not observable, but what we can observe is whether the plant is whether the plant is tall or dwarf. Okay, so this is the phenotype. So this that is the this is the phenotype, that is the observable character, and this is the genotype. So if we go by the definition, genotype it is the specific set of genes that an offspring inherits from its parents, while phenotype is the observable characteristics such as physical traits, behavior or development which result from the interaction of this genotype with environmental factors. Okay, now let's take the example, like let's take another trait for an example. Suppose flower color. Okay, suppose we are taking the example of flower color. So this is the character and it has two traits, that is purple and white. So these are both like genetically coded, like these are alleles to each other, right? So if we refer to purple as capital P, we'll refer to the white as small p. Okay, now Mendel made an interesting observation. He saw that purple was appearing in the population, like there was maximum appearance of purple in the population. Okay, this was maximum, and there was a lesser occurrence of white flowers among the population. Okay, so on the basis of occurrence of a trait, the allele can be classified into three types. Okay, I mean, on the basis of occurrence in the population, like occurrence of trait in the population, allele can be classified on the basis of three types. So on the basis of occurrence in population, alleles can be classified into three types. Number one is the wild allele. Okay, that shows maximum population. Number two is the polymorphic allele. Okay, these are alleles present in alleles present in more than 1% of population. And third is the mutant allele. Okay, third is mutant allele, okay, present in less than 1% of the population. Okay, suppose if we take a population for an example, okay, let us say there is an area, and this is the occurrence of a like alleles. Okay. Okay, so we can say from this that, see, the wild allele says maximum population, so here we can observe that the population of A is maximum, so A is the wild allele. Okay, then you can see there are like 3 small a's, and here the definition says that polymorphic alleles are alleles present in more than 1% of the population. So small a will be the polymorphic allele, while you can observe that there is only one occurrence of this alpha allele. So alpha can be referred to as the mutant allele. How an allele is expressed in the morphology of an organism, alleles can be classified on the basis of that also. So on the basis of expression, alleles can be classified into 2 types, that is dominant allele and recessive allele. So on the basis of expression, alleles can be classified into 2 types. Okay, one is the dominant allele, another is recessive allele. Okay, so it is generally referred to in written in capital letters, like capital letters are used to express them. Suppose for the plant character of flower color, so the 2 traits are purple and white, right? So purple flowers, the allele for purple color of the coloration of the flower is dominant over the white coloration of flower. Okay, so example, like flower color for this character, purple and white. So purple is the dominant allele. Okay, so it is referred to as capital P, and this one is referred to as small p. Okay, and the major thing we should keep in mind is that it is present both when it's present as homozygous condition or heterozygous condition. Okay, so it is expressed in both homozygous as well as heterozygous condition. For example, suppose both P and P alleles are present, so the plant will show purple color flowers. Okay, purple flowers, as well as if the P and capital P, that is one allele for the purple flower phenotype, and another for the white color flower phenotype is present. So it will still show, it will still have purple flowers. Okay, so keep in mind here the capital A allele is for the flower phenotype. Okay, now now comes the recessive allele. Okay, so what are these recessive allele? So recessive allele is that they are generally referred to as generally demonstrated in small letters. Okay, they are generally demonstrated in small letter. That is example the allele for the white flowers in white color flower in pea plant. The allele is referred to in as small p. Okay, and another thing you must keep in mind is that they are expressed only when they are present in homozygous condition. Okay, like this is the major thing you should keep in mind, that it is only expressed only, this is the word, only expressed in homozygous condition. That means when two alleles, suppose for small p and small p are present, then only the pea plant will show white color flowers. Okay, not when capital P or small p. This time they will show purple flowers, but only when the plant has these two alleles, like if the alleles for both the flower colorations are small p and small p, that is the recessive, both are recessive alleles, then only they will show the recessive trait. So to summarize this, we can say that also one thing you must keep in mind, that allele should always be written in pairs. Okay, like if you are referring to as tall, like there is the, to refer the genotype of an allele, you should always write in pair. Okay, it's like for tall you can write as TT or TT, right? Or for like dwarf it will be small t small t. Okay, like this should always be written in pairs. Okay, so to summarize this, each parent diploid has two alleles for a trait, so they may be homozygous dominant, it is two dominant alleles for a trait TT. Okay, they may be homozygous recessive like two recessive alleles for a trait, or they may be heterozygous, like the genotype may be heterozygous, that is one of each allele for a particular trait. Okay. Now, before we study Mendel's laws of inheritance, let's learn about what is the meaning of crosses and what are the meaning of monohybrid, dihybrid, trihybrid crosses. Okay. So, in genetics, a cross may be defined as the purpose of meaningful mating of two different individuals to study the inheritance of genetic material, producing offsprings with a combination of their parents' gene. Okay. So, what is the cross? Cross is purposeful mating of two different individuals to study the inheritance of genetic material producing offspring with a combination of their parents' gene. Okay, so this is the meaning of cross. Now we have we'll mainly consider three types of process, but it can be tetrahybrid cross as well. The first is the monohybrid cross. So in monohybrid cross is a crossing between two plants where only one character is taken under consider. Okay, only like one character is taken under consideration, like a tall plant and crossed with a dwarf plant. Okay, here is an example. Like next is dihybrid cross. So here we take only two characters are taken under consideration. Like suppose we are taking two characters like round seed, purple flower, like a plant having round seeds and purple flower is crossed with a plant having wrinkled seeds and white flower. Similarly, we have trihybrid cross, where only three characters are taken under consideration. Okay, like example, plant bearing round seed, purple flowers, and tall plant is crossed with a plant bearing wrinkled seeds, white flower, and it's dwarf. Okay, now let's come to the laws of inheritance. Okay, this is a very important part. So laws of inheritance. So this was proposed by Mendel in the 1800s, but rather late 1800s, but were accepted in early 1900s. And one thing you should keep in mind, that it's very important, these laws are only applicable on sexually reproducing eukaryotic organisms. Okay, so they're applicable on sexually reproducing eukaryotic organism, not on prokaryotic or others. Okay, so initially he proposed 3 laws: law of dominance, law of segregation, and law of independent assortment. But later this law was not accepted, and now we have only 2 laws. Like this can be considered Mendel's first law, and this is Mendel's second law. We will learn about them in details in the next class. Thank you for joining the class and have a great day.